Hi everyone, welcome to my channel Srinivasa Biology Tutorial. For this session to discuss NCERT class 11 cell cycle and cell division part 3, that is meiosis. What is meiosis? The production of offsprings by sexual reproduction including the fusion of two gametes each within complete haploid set of chromosomes. So here any one parental cell to divided into in a meiosis division to forms how many cells? 4 haploid cells to get produced. To get forms as how many? 4 haploid gametes. Haploid gametes. The gametes formation of the specialized diploid cell. So these gametes, how do they come with it is a diploid cell to get producing the haploid cells. Here this specialized kind of the cell division is reduce the chromosome number of result in the production of the haploid daughter cells. Hence this kind of division is called as in a meiosis, or it is called as in a completely what? Reductional division. It is what? Reductional division. So here see what happened, the chromosome will be there, chromosome number there awfully means there are half of chromosomes to get distributed to as in a the parental cell to as in a daughter cells. So this process is called as what? Meiosis. Now the meiosis ensures the production of the haploid cells. Here the production of what? Haploid cells. The haploid phase of the life cycle, the sexual reproducing organisms, whereas in the fertilization are restored the diploid phase. The diploid phase of the cells to restore to get forms as in a haploid cells. Now we come across the meiosis during the gametogenesis in plants and animals. So this gametogenesis mostly takes place in the plants and animals. Leads to the formation of the haploid gametes. The key features of meiosis they are followed. Some key features they given. Now see that one by one. Now the meiosis involved that in the sequential cycle, that is in a first one is nuclear and nuclear division is called as in a the involves the two sequential cycles. Here one is the nuclear division and second one is cell division. In the process the first meiosis and meiosis 2. Meiosis 1 and meiosis 2. So in this meiosis 1 and meiosis 2, but only one single cycle DNA replication takes place. Here how many phases present? The two cycles as present. One is a meiosis 1 and second one is meiosis 2. But DNA replication takes place only one phase. The meiosis 1 is the initiate after the parental chromosome have the completely the replicate to produce the identical sister chromatids of the S phase. Here the meiosis 1 that initiated after the parental chromosome, they having as what? To produce as in one chromatids in a only S phase, in that S phase. Now in this meiosis involves the pairing of homologous chromosomes and the recombination between the non-sister chromatids, homologous chromosomes. So in this meiosis what happened? Here the chromosomes, see these are the chromosomes, am I right? These chromosomes completely what happened? They are crossing over, they are completely crossing over the external genetic material on the two non-sister chromatids. Here for 4 haploid cells are formed as an end of the meiosis 2. So the 4 haploid cells to get formed end of the in a meiosis 2. Now the meiotic events can be grouped under the following phase. So they given as in a 4 phases, like as in a meiosis 1 to divide as in a 4 phases, meiosis 2 also to divide as in a 4 phases. They are prophase 1, metaphase 1, anaphase 1, telophase 1. And at the same time in the meiosis 2 also prophase 2, metaphase 2, anaphase 2 and then telophase 2. Now you will discuss as in a one by one. First we discuss as what? In a meiosis 1. The first one is what? Meiosis 1. In this meiosis 1, they are followed as in different phases. They are first one prophase 1. Prophase 1. The prophase 1 is the first meiotic division. A typical longer phase. It is a longer and more complex when we compare to as in a prophase of in a mitosis. So it is in a longer one as well as in a very complex when you compare to as in a mitosis. When you compare to as in a mitosis, it has in further subdivided into as in a following 5 phases. Again to divide how many phases? 5 phases. They are leptotin, zygotin, pachytin, diplotin and diagenesis. Here, based on the chromosomal behavior, based on what? Based on the chromosomal behavior based on, they divide as in a leptotin and then zygotin, pagatin and diplotin and diakinesis. Now see one by one. In this, during the leptotin stage, what happened? The leptotin stage, the chromosome becomes gradually visible under a light electron microscope. You see that in under electron microscope, the chromosome gradually they are appear like gradually to get appear like. Now, the compaction of any chromosome continues through the leptotin. This is followed by as in a second stage is prophase. One is called as in a zygotin. So after in this phase, we see that in a second phase is what? Zygotin. In this, during this stage, the chromosomes start the pairing together, and the process is association with called as in a synapsis. So in the second phase, what happened? In that second phase, here the chromosomes, here see, the chromosomes are completely, they forms as what? Synapsis. To get forms as what? Synapsis. See, synapsis means what happened here? See, the chromosomes to get forms as in a synapsis like. They start after pairing, they get forms as in a synapsis. Okay, now the paired chromosomes are called as in homologous chromosomes. When they get forms like these, chromosomes are called as what? Homologous chromosome. The electron micrograph of this stage indicates that in a chromosome synapsis they are accompanied by as in a formation of complex is called as a synaptomal complex. So when this completely they comes as a nearby, this complex is called as what? This is in a synaptomal complex. It's called as what? Synaptomal complex. This complex formed the pair of the synapses homologous chromosomes is called as in a bivalent. See, here to get forms as in a bivalent chromosomes is present, or nothing but as in tetrads, or nothing but as what? These are the tetrads. Okay. However, these are in more clearly visible the next stage. So these are completely clearly visible in that next stage. The two stages, the prophase one, the relatively short-lived, the compared to as the next stage of the pagatin. So when you compare to as in a pagatin, it is in very short-lived. Next, here during this stage, the four chromatids of each bivalent chromosomes, it is distinct clearly appear as a tetrads. Mostly clearly they appear as in a tetrads. Now, here chromatids, the bivalent chromosomes become distinct and clearly they appear as a tetrads. Now this stage, the characterized by an appearance of the recombinant nodules, the site of the crossing over between the non-sister chromatids of the homologous chromosome. So here, for example, I am taking as this homologous chromosomes. So in these two homologous chromosomes, they are completely the crossing over. Here they are completely crossing over. Here to get form as the recombinant nodules. Recombinant nodules to get form as what? Recombinant nodules. Now, the crossing over the exchange of genetic material between the two homologous chromosomes. Crossing over also enzyme they are mediated is present. That enzyme is the recombinase enzyme. Here, which enzyme they are involving here? Recombinase enzyme. Very, very important. Recombinase enzyme is involving. Here, recombinase enzyme is involving. Now, after the crossing over leads the formation of the genetic material of the two chromosomes. So the total chromosomes what happened to get completely exchange the genetic material. The recombination between the homologous chromosomes is completely their end of the pagatin, the leaving the chromosomes linked site of the crossing over. So here what happened? This for example is the A chromosome, it is in a B homologous chromosome. So A to B, B to A, the genetic material will be there. Exchange was takes place in the pagatin stage. Next last one. What is that one? Pagatin. After this pagatin, now to discuss in a diplotin. In this diplotin, what happened? In this diploidine, recognized as in the dissolution of the synaptomal complex and the tendency of the recombinant homologous chromosomes bivalent that separate to as each and a chromosome except to as a sister the sites of the crossing over these X-shaped structures is called as in a chiasmata. So when this completely their chromosomes are separate, when they get chromosomes are separate, what happen? They appear as in a X-shaped chromosomes. They appear as in what? X-shaped. See, they appear as in a X-shape. This is called as what? This is in a chiasmata. The oocytes of the some vertebrates and the diploidine can last for the in a months or years. In some vertebrates, the diploidine can be lost for as in a months or years also. The last final stage of the meiotic prophase one is called diakinesis. The diakinesis is marked by as in a the termination of the chiasmata. So the finally what happened? They get completely separated after the separation of their chromosomes to get forms like. After they are separated and during the phase of the chromosomes are in finally the fully condensed and the meiotic spindles are their assemblage prepare for as in the homologous chromosomes for the separation. The homologous chromosome separation takes place. By end of the diakinesis, the nucleolus disappear and the nuclear envelopes also the breakdown and the diakinesis the transition of the metaphase. So here what happened when this nucleus and nucleus will be disappear, what happened? Then enter into as in a metaphase. Here in this metaphase what happened? Already you know the metaphase. Here the chromosomes comes as in a metacentric plate, right? See in the metaphase 1, the bivalent chromosomes aligned as an equatorial plate. The microtubules their opposite poles to as in a spindle attachment of kinetochore homologous chromosomes. So here in this metaphase what happened? Two ancestors will be to get formed here. Then here the chromosomes comes as in a completely metacentric plate. It is in a metaphase. And next anaphase what happened? See in that anaphase what happened? The homologous chromosomes are separated while the sister chromatids are remain there associated with as in a centromere. So the total chromosomes will be there pull up and to get reached as in a end of the region. Next telophase 1 what happened? The nuclear membrane and nucleolus and reappear. Again they are formed as a nuclear membrane, nuclear envelopes, and here the cytokinesis followed by as in is called as in a dyad cells. So after the cytokinesis to get forms as in a two double cells. Okay, so that is next all through in many cases of the chromosomes to undergo some in a dispersion. Means after their chromosomes will be to undergo, they again to divide. They do not reach the their extremely the extended state of the interface of nucleus. These are not formed as any interface of the nucleus. Now, this stage between the two mitotic divisions is called as an interkinesis. To the cytokinesis and karyokinesis between the division, in that mitosis and meiosis between this division is called as what? Interkinesis. Is called as what? Interkinesis. And generally it is short-lived. It is generally short-lived. This is a no replication of DNA during the interkinesis. Interkinesis followed by as a prophase 2, much simple prophase than prophase 1. So in this prophase 1, how does it takes place? Not like, but similarly, that kind of the divisions was takes place in the prophase, in meiosis 2. Okay. Now see in the meiosis 2, what happened? In the meiosis 2, again they have as a name prophase 2, metaphase 2, anaphase 2, telophase 2. Now see the first one is what? The prophase 2. The meiosis 2 is the initiated immediately after cytokinesis, usually before the chromosomes have fully elongated. So in this prophase 2, what happened? The chromosomes fully they get elongated. As well means condensation process takes place. It's considered as a meiosis 1, meiosis 2. The resemble the normal the mitosis. It is equal to as a normal mitosis. The nuclear membrane they disappear end of the prophase 2 and chromosomes all gene becomes their compact. The compact condensation takes place in the prophase 2. Next metaphase 2, what happened? This stage the chromosomes are in the equatorial and the microtubules form opposite poles. So the spindle get attached to the as a kinetochore of the sister chromatids. So the chromosomes to get attached as in a within a central kinetochore, to get attached with as in a like same in a central kinetochore like. Okay, next anaphase. Anaphase to what happen? Ana means up, yes na. So within the DNA simultaneously the splitting of the centromere. So when they get pull up, what happen? They get separated. Their centromere, each of the chromosomes here allow as a move upward opposite poles of the cell. So they get both are they comes as an a opposite poles. The short timing, the microtubules attached to as an a kinetochore. So here the kinetochore region to get attached to as an a chromosomes. Next last one is in a telophase 2. In that telophase 2. In this telophase 2, what happens? See, they given clearly here. The prophase 2, metaphase 2, anaphase 2, and then here telophase 2. Okay. Now, in this anaphase, the telophase 2. The telophase 2 is the last phase of the meiosis 2. Clear? Now, here what is the telophase 2? See, in this telophase 2, the meiosis end with as a telophase 2, which is the two groups of the chromosomes. One is again to get enclosed as a nuclear envelope. So, nuclear envelopes to again to get formed. The cytokinesis for to result in the formation of the tetrad cells. So, after to get formation of the the karyokinesis immediately following as in what? Cytokinesis. So, then what happened? Four tetrads will be to get formed. So, the daughter cells will be to get produced. So, this is incompletely the telophase 2 of meiosis 2. Okay. Now, what is the significance of the meiosis? See, now the significance of meiosis is in a mechanism which is a conversion of the specific chromosomes and number of each species of achieved across generations of the sexually reproduction organisms. So, sexual reproduction organisms, they complete their followed by this meiosis. Even through the process, the person and the paradoxically as well as result the reduction of the chromosome number by half. So, in this process, what happened? Their reduction, the chromosome number will be the reduction. This takes place and their chromosome number to get distributed as in daughter cells. Next, it is also increasing a genetical variability in a population of organisms from as in one generation to next generation. The genetic information to get passed from one generation to next generation. The variation are in very important for any process of in evolution. So this total variation is very important for as in the next generations. Means evolution development purpose it is very important. Okay, this is the significance of meiosis. Okay, this is the completely the cell cycle and cell division chapter. Okay, please subscribe, share and like. Thank you for watching.